Okay, take it away, Jen.
Okay. Thank you so much for having me. I gave a talk to the Westchester County Bird Club back in 2019 as a first year graduate student. So it's really exciting to be back and show you all the work that I've been doing over the past 3 years at Cornell. So today I'm going to be talking about microbiota-mediated behavior in wild birds. So I am a very proud first generation college student. So I'm the first in my family to attend a 4-year institution. And I'm from a really small town in Juniata County, Pennsylvania called Mifflintown. And when it came time to go to college, I wanted to stay close to home, but still go far away enough that my mom couldn't come visit every weekend. So I decided to go to Millersville University in Lancaster, where I thought that I was going to become a medical doctor. So in high school, I loved biology. And I feel like if you're good at biology in high school, everyone tells you you should go to medical school to be a human doctor. So my first year of college, I took a human and anatomy course and I saw a cadaver for the first time. And when they pulled the sheet back, I fainted and hit the floor. And so my ambitions of becoming a medical doctor quickly went away. So I had to pivot to something else. So I really liked tutoring other students in high school, and I started doing the same at Millersville. So I decided to switch into the secondary education track and become a high school biology teacher. But then in my introductory lab, I got the opportunity to hear the word research, and I didn't really understand what that meant at the time. But there was a new professor, Brent Horton, who wanted to study birds in the Ecuadorian Amazon. And I had never been outside of the country before. So I decided to take a leap of faith and see if research worked out for me. And then I had the amazing opportunity as a sophomore in undergraduate to travel to Ecuador to work on this beautiful bird here called the wire-tailed manakin. And I subsequently went back to Ecuador a total of 3 times. And that was really one of the most influential experiences for my decision to continue studying ornithology. So this is a male wire-tailed manakin, and manakins are a clade of birds that have these really elaborate courtship displays. So males will go around and they dance with each other and they try to impress the females. And I remember I was at a conference when I was in undergrad, and all these manakin species, they have these elaborate displays where they're doing kind of loops over each other and all these acrobatics. And someone that was studying the black manakin thought that the bird was just doing a really boring display. So the black manakin, it looks like it's just jumping up and down on a branch. But then someone decided to film the bird and play the video back really slowly. And when they played it back frame by frame, they saw that the black manakin was actually jumping up in the air and doing a backflip faster than the human eye can see. So that's just an extreme example of the kind of elaborate courtship displays that these birds do. So I had never really thought about birds before. I probably couldn't even tell you the difference between a cardinal and a robin. So when I went to Ecuador, I got to work with birds for the first time. And I learned how to capture birds with mist nets, mark them with color and metal bands, take samples such as blood samples to measure their hormones, and also how to measure them to assess their condition and health. So one of the most amazing parts of being in Ecuador was the extreme amount of biodiversity that I saw, not just in birds, but across animal taxa. And this butterfly here is a blue morpho butterfly. And this was a really surreal experience to have one land on my hand, and I was able to get a video of it. This is an earthworm that I really had to talk myself up to pick it up, but I'm glad that I have the picture now. And then even though we were targeting wire-tailed manakins with our mist nets, we also caught a variety of other species. And this beautiful guy over here with the fan, that is a royal flycatcher, very photogenic. He looks like he's smiling. And then on the bottom row from left to right is a red-rumped arisari. When I reached into the bag to grab that bird, it got me right here. So I always remember that bird. And then a yellow-billed jacamar and a blue-crowned manakin. So then after going to Ecuador my first year and I held my first bird, I knew that I wanted to pursue ornithology for a career. And I wanted to test out whether I wanted to do more basic research where I'm just interested in answering cool questions about nature, or if I wanted to do more applied conservation work. So the following summer, I applied to be a conservation science intern at Hawk Mountain Sanctuary in Kempton, PA. And I spent the summer there doing a variety of projects on birds of prey. For example, on the left here, we would go out and meet with local landlords to look at the kestrel nest boxes that were on their properties and go up and measure the nestlings. I got to tag black vultures. This is one of my favorite pictures of myself because black vultures are really crazy birds whenever you're trying to catch them. So I was very proud. You can tell by the way I'm smiling. And then I also got to do work with broad-winged hawks by going on nest searches and capturing them and putting radio transmitters on their back. So with those two experiences, I knew that I wanted to pursue research as a career, and especially on birds. So in 2018, I graduated from Millersville with my bachelor's in science and animal behavior. And then I applied broadly, but Cornell with the Lab of Ornithology here is one of the top places to study birds in the world. So I found out that I got into Cornell's Ecology and Evolutionary Biology PhD program. And now I'm currently in my last year of my PhD program, looking to become a professor after this. So I made the transition from working on manakins and raptors. So what am I doing now at Cornell? So by working in Ecuador, the project looked at the effect of hormones on social behavior in male manakins. So I knew that I wanted to work on hormones. I also love the elaborate courtship behaviors that were conducted by the two males. So I wanted to incorporate behavior into my research. And then I heard about this crazy thing called the gut microbiome or the gut microbiota. So what is the gut microbiota? And I kind of meshed all of these things together. And now my dissertation at Cornell is looking at microbe-mediated behaviors in wild tree swallows. So first, let's take a step back and define what the gut microbiome is. So whenever I think of the gut microbiome, or I ask my family and friends, they always refer to different things that they've seen on social media and in grocery stores. So recently, one thing I've seen is that Dove body wash is actually now calling their body washes microbiome gentle for the microbes that are on your skin. And also, if you're walking through the grocery store aisle, people always say you should eat yogurt for the healthy microbes in it. So you may be seeing things such as this Activia probiotic. But the formal definition of the gut microbiome is all of the microorganisms. So this includes bacteria, viruses, archaea, fungi, and protozoa, and their collective genes that are residing within the host gastrointestinal tract. And the gut microbiome has received a lot of attention in recent years because of all of the beneficial roles it plays for the host. The first being nutritional uptake, specifically looking at a nutrient called short chain fatty acids. And when we as humans ingest dietary fiber, we don't naturally make the enzymes that are used to digest this really complex fiber down to digestible units. So we actually have bacteria inside of our guts that help break it down through a process called fermentation. And then it produces these smaller molecules called short chain fatty acids that then we can absorb and use. In addition to nutritional uptake, gut microbes also help with detoxification and waste removal. Specifically, this example is about indigestible plant defense compounds. So this is a really cool bird called a hoatzin. And the hoatzin is one of the only birds that almost exclusively eats leaves. So when these birds are ingesting these leaves, some of their prey items have these really toxic plant compounds that plants use for defense. So the gut microbes inside of the hoatzin gastrointestinal tract are specialized to help digest these defense compounds and inactivate them so that the bird can continue eating the plant. And finally, in addition to nutritional uptake and detoxification, the gut microbiome is also really important for training the immune system. So whenever we're born, we acquire a certain gut microbiome, both from our mother and through the environment.
Jen, can we interrupt?
Yeah, sure.
Sorry, go ahead.
Yeah, I guess since we can't hear that well, that hoatzin bird, did it, is the plant got some kind of something to make it sick, and so it has to toxin? Does it have a toxin that the bird gets rid of?
Right.
What is the plant that it eats?
That is a great question. I don't know specifically what species of plant it is, but just in general, any kind of plant that has a toxin used as a defense mechanism, there is a species of microbe inside of the hoatzin that is able to deactivate those. Can you hear me okay? Is that better if I speak closer to the screen?
You're good.
Actually, and can I just say one more thing?
Sure, yeah.
You don't need to, this is about trying to get the speaker to work.
Yeah.
This doesn't need to be on Bluetooth because you have it connected to the computer. So if you have it selected, it should work.
I know, but it dropped it.
Oh, why don't you just try one?
We can.
We're just trying it one more time.
Yeah, no problem.
We're recording this. No, it's not detecting.
Oh, all right. Go ahead. Keep going. I'm sorry, Jen.
No,
And
that's
thank
okay.
you for helping me understand that plant.
Oh,
thing.
yes. No, feel free to, I have the Zoom window muted, but feel free to unmute yourself. And if something is not clear, I'm happy to take a question right away. Okay. So yeah, as I was saying, whenever we are born, we acquire a certain gut microbiome, both from our mother and from the environment. And the gut microbes actually help train our B cells. B cells are what produce antibodies that then attack pathogens. So the gut microbes actually train our immune system what to recognize as a pathogen so that we don't develop autoimmune disorders where our immune system is attacking our own cells. So with that, we know that the gut microbiome influences a wide range of host traits, including metabolism of complex diet items, detoxification of plant toxins, which then are used and removed from the system as waste, and also immune function. But there is little work on if the gut microbiome can actually influence host behavior. So my work is investigating how gut microbes may be producing molecules that then travel up to the host brain and influence behavior. And then also vice versa, where the way that animals are interacting with each other may influence their own gut microbiome diversity and composition. So here are some examples of how microbiota can influence host behavior. In spotted hyenas, they have a scent gland that they use to produce a unique volatile signature that then others can sense. And we find that the scent gland microbiota actually co-varies with reproductive status. So male spotted hyenas are able to smell these volatiles that are produced by the gut microbes, and that indicates when females are ready to reproduce. In addition, in red harvester ants, they have microbiota that are on their exoskeleton, and their external microbiota also produce these kind of volatiles that influence nest mate recognition. And then finally, in fruit flies, the microbiota influence mate preference because they, again, are producing these sex pheromones that males and females can recognize. And then if we flip it around to look at how host behavior can influence the gut microbiome, in yellow baboons, we know that grooming partners, so where baboons are kind of picking all of the insects off of each other's backs through their fur, we find that the grooming partners have a more similar gut microbiota because they interact more. In the viviparous lizard, they have what's called a polyandrous mating system. So this means that one female will mate with multiple males, and females that mate with more males have an increased microbial diversity, so more microbial species. And then finally, in black-legged kittiwakes, we know that the birds that are mates have a more similar cloacal microbiota. So with all of this background research, I decided to attend Cornell, one, because of all of the bird knowledge that's already here, but also the current lab that I'm in is Marin Vitusek's lab, and we study tree swallows. And there has been an active population in Ithaca, New York for the past over 35 years. So we have about 500 nest boxes that are set up around Ithaca, where we have a long-term population that we monitor year to year. And one of the really neat things that we have to measure behavior is, instead of using traditional binoculars, we can actually use technology to monitor behavior remotely. And the way that we do this is we have something called an RFID reader, or a radio frequency identification device. And this is just basically a mini computer that has a memory card that then we attach to the bottom of the box, and it's powered by a little battery. And then what we do is we wrap an antenna around the entrance of the nest box. And tree swallows are cavity nesters, so they exclusively will nest inside of old cavities in trees, or in this case, any artificial nest boxes that we put up around water. And each summer, when we catch a bird, we will mark it with a metal band that has a unique ID, but on its other foot, we put something called a pit tag, which is a passive integrated transponder tag. And this is basically a unique electric code that's associated with each pit tag. So then that's attached to the bird's band. And then whenever we have birds that fly through the entrance hole, the antenna around the nest entrance hole actually picks up who it is and how often they're visiting. So we're able to track things such as nestling feeding rate and social networks by looking at the unique pit tag IDs. So the whole point of my dissertation is to assess whether there is a bidirectional relationship between the gut microbiome and host behavior. So first, we'll look at how microbiota affects host behavior. And across vertebrate taxa, we know that timing of breeding is a major determinant of organismal fitness or success in nature. And across seasonally breeding birds, we find a relationship where earlier breeders have higher reproductive success. So here on the bottom axis of this graph is timing of breeding, or lay date of the first egg. And on the y-axis is reproductive success, or how many offspring they produce. And again, we find this inverse relationship with the birds that are arriving to the breeding sites first are having increased reproductive success. But why is this the case? It could be that the birds that are arriving back earlier get first choice of the high quality mates. They get access to the best nesting sites, such as nest boxes that are close to water. Tree swallows are aerial insectivores, which means that they exclusively eat aquatic insects that emerge from water. So we find that the birds that are nesting right beside water often have the most nutrition. Or it could be that if the bird arrives back early and for some reason their first clutch fails, they have enough time then throughout the rest of the breeding season to re-nest and raise a successful brood. So again, in tree swallows, we find that earlier breeders have higher reproductive success. So here on the x-axis, or the bottom axis of this graph, is average week of clutch initiation. And then on the y-axis is lifetime number of fledglings produced. And tree swallows are a really great system to work with because the females also have high site fidelity, which means that if a female comes to breed at a site, they'll continue to come there year after year. So we're able to monitor lifetime reproductive success. So here again, we find that the tree swallows that arrive back to the breeding site earlier produce a higher number of fledglings throughout their lifetime. So if breeding earlier increases overall fitness and success, what prevents all individuals from breeding earlier? And in Ithaca, New York, our early springs are riddled with harsher and more unpredictable weather events. It's really common for me to find snow inside of the nest boxes in the middle of May sometimes. So some individuals are able to withstand these early season conditions while others can't. And one hypothesis is that individuals that are able to change certain aspects of their phenotype, the phenotype is just any measurable trait that you could find in the bird, such as plumage color or mass or anything that you can observe with the human eye. So to demonstrate this, on the x-axis or the bottom axis, we have the different stages of breeding, such as incubation and nestling provisioning. And on the y-axis is some measurable trait. So birds that remain static in some aspect of their phenotype will have no change from incubation to provisioning, but then others may actually change certain traits in response to the different demands of each stage of breeding. And in female tree swallows, we know that early breeders change their body mass over time. So the early breeders arrive to the field sites heavier during incubation. And then by the time they begin provisioning young, they lose the majority of their body mass. And it may be better to be heavier during incubation so that you can sit on the eggs for longer bouts of time and produce more heat. And then by the time you begin provisioning young, it's better to be lighter so that as you're flying and making all these feeding trips, it's more energy efficient. So overall, we know that the earlier breeders have higher reproductive success, but we don't actually know what part of their physiology is mediating this adaptive mass loss across the breeding season. So one hypothesis could be the microbiome. Because the microbiome is involved in digesting really complex diet items, it could influence things such as body mass. And for example, a tree swallow may arrive to the field site with a particular microbial composition with different species. And then as the breeding season progresses and demands change, their gut microbiome may shift in a certain way, such as reduced diversity or a different composition of species. And this could result in either no change at the organism level, or we could see that a change in the microbiome also causes a change in some traits of the host. So to test this, back in 2017, we caught female tree swallows, the same individuals 3 times throughout the breeding season. And the first capture being on day 6 of incubation. And then we took a cloacal swab to sample the gut microbes. And then we caught them again on day 3 of nestling provisioning, where we took another coliacal swab. And then on the third day, or on the third capture, was day 7 of nestling provisioning. And then we do some fancy stuff with DNA and through sequencing, and we eventually go from just this swab to being able to differentiate what microbial species are actually there present inside the bird. So the first question that we have is, what does the diversity look like when the birds are arriving at the breeding grounds? So here on the bottom axis, we have clutch initiation date, or the day that the first egg is laid. And on the y-axis is microbial diversity, measured as the number of species. And we find this really interesting relationship where the females that are breeding earlier have higher diversity at incubation. So this could be an indication that having more microbial species is associated with being able to withstand these really harsh early season conditions. And then in addition to looking at static points of diversity at each stage of breeding, we can also look at the change in microbial diversity from between each capture. And so here again, on the bottom axis, we have clutch initiation date. And now if you notice on the vertical axis, we have change in microbial diversity. And the earlier breeders aren't just arriving with a more diverse microbiome, they're actually also losing more diversity between incubation and early provisioning. And then finally, we want to assess how this change in microbial diversity may be correlating with adaptive mass loss. And here on the x-axis, or the bottom axis, we have change in body mass between incubation and early provisioning. And again, on the y-axis, or the vertical axis, we have change in microbial diversity. And we find this really interesting positive correlation where birds that lose more body mass are also losing more microbial diversity between incubation and early provisioning. So to summarize, we know that earlier breeders lose more mass between incubation and provisioning. The early breeders are also losing more of their gut microbial diversity between these two stages, and they have high reproductive success. So there's this really interesting relationship where microbial diversity and flexibility, or the ability to change your microbial diversity, are associated with lay date in female tree swallows. And again, just to summarize, we know that early breeders have increased reproductive success. They're also losing the majority of their microbial diversity between incubation and nestling provisioning. And they're also losing the majority of their body mass between incubation and early provisioning. So there is this positive relationship with microbial diversity loss and body mass loss, but this is all correlational. So the directionality and the causality of this relationship still needs to be tested, but it's given us some super interesting findings to follow up on in future studies. Okay, so now we will assess whether host behavior affects the microbiota. So flipping to the other side of this bidirectional arrow. So again, we're going to study this in tree swallows, and female tree swallows actually exhibit natural variation in sociality. So as I was saying before, there is a population in Ithaca, New York that's been studied for over 35 years. And we have a grid system of artificial ponds that we have hundreds of wooden nest boxes set up. So you can see how all of the nest boxes are placed at least 10 meters apart. Tree swallows are semi-social, so they still battle each other sometimes, but through our technology that we put on the boxes, we know that all the birds are visiting each other, and We don't actually know what's going on yet, but our next step is to put GoPro cameras actually inside of some of these boxes to assess whenever our RFIDs are picking up who it is, to actually tell what kind of behavior is going on. So in female tree swallows, the brightness of the white breast plumage predicts the degree of sociality in females. So females that have a brighter white breast plumage are more social. So to test this, we did a really high-tech experiment where we wanted to manipulate some social signal that the birds use to alter their behavior. So what we did was we took a gray marker and we dulled their white breast plumage. So this is just an example of what it looks like. It was always pretty funny to watch. But then for another group of birds, we wanted to make sure that the scent of the marker didn't have any effect. So in another group of birds, we actually colored them with a clear marker just to equalize disturbance across the treatment groups, and also to make sure that it wasn't the smell or some other aspect of the marker that was influencing behavior. And then again, we sampled birds 3 times throughout the breeding season. The first capture being on day 6 of incubation, second capture being on day 3 of nestling provisioning, and then the final capture on day 7 of nesting provisioning. And then at the first and third captures, we did the dulling or the control treatment with the different markers. And again, to sample the gut microbiome, we took a cloacal swab before and after the coloring. Okay, so again, we find some really interesting results based on our RFID system that monitors social behavior. So the birds that received the dulling treatment, where they had their bright breast plumage dulled with a gray marker, we find that the dulled females are visited less frequently by the male tree swallows. So by dulling the treatment, we find that the males aren't as interested in the dulled females. And then again, we can look at the amount of times that the dulled females are visited by other females. As I mentioned before, we find these really interesting social networks where female tree swallows are going and they're visiting each other's boxes. And again, we're not really sure what's going on yet, but we find this really interesting results where the dulled females are visited more frequently by other females. So again, the other females are assessing that the focal female has duller breast plumage, and they're going and visiting their nest box more frequently. And we also wanted to assess how the dulling treatment impacted the microbial diversity as well. And we find that the females that are dulled have a higher cloacal microbiome in comparison to controls. So we see this increase in microbial diversity in the birds that receive the dulling treatment. Okay, and finally, the whole point of being a bird and breeding is so that you can raise offspring and fledge them. So we wanted to assess if this treatment had any effect on their reproductive success. And again, we find at different stages of breeding, including hatching success of the eggs, survival of the nestlings until day 12 of development, and then whether or not the offspring have actually successfully fledged the nest, we find that the dulled females have higher numbers of nestlings. So overall, this dulling treatment actually increases reproductive success. So that was a lot of information thrown at you. So to summarize, we conducted this color manipulation and it caused changes in behavior and subsequently microbiome diversity. So the birds that received the dulling treatment with the gray marker, they had altered social behavior, whereby the adult females had an increased visitation by other females and decreased visitations by males. For their physiology, the adult females had an increased microbial diversity. And then for reproductive success, we find that the adult females have increased hatching success of their eggs and also increased fledging success of their fledglings. So if we take a step back and we look at this overarching question again, is there a bidirectional relationship between gut microbiota and host social behavior in tree swallows? And yes, we do find a potential relationship between microbiota and timing of breeding. So yes. And then we assessed whether manipulating a social signal such as the white breast plumage, if that influences the gut microbiome composition. And yes, it does. So there is evidence for a bidirectional relationship between gut microbiota and social behavior in tree swallows. And some future directions. So because this relationship between microbial diversity and mass loss was just based on descriptive work and going out and sampling the birds without any kind of experiment, we want to disrupt the adult microbiome using antibiotics. So one way that we can do this is that when female tree swallows and any bird that is incubating, whenever they are preparing to sit on their eggs, they lose all the feathers on their breast and develop something called a brood patch. And that's pictured here in this photo. And so they lose all the feathers, and then that piece of skin becomes really highly vascularized with blood vessels. And this is to help give more heat to the eggs, because then you'll have direct skin to egg contact. And one method that I've developed during my PhD is that you can take an antibiotic and you can dissolve it inside of a gel. And then we put this gel that has the antibiotic in it on top of a fake egg that is the same shape, size, and color as a real tree swallow egg. We place this fake egg inside the nest. And then when the female bird is incubating on top of the egg, I've been able to show that the antibiotic dissolved in the gel actually diffuses up through the skin and is able to systemically alter the gut microbiome. So I am currently analyzing the results of that experiment, so stay tuned. But the whole purpose of this is to test the causality of this relationship that we see with this graph here, where on the bottom axis, we have female microbial diversity, and on the y-axis is offspring fledged. So again, we find this positive relationship between microbial diversity and reproductive success. So this experiment was testing whether if you disrupt the gut microbiome, does it influence their ability to fledge their offspring? And then taking a step back from the nitty gritty of data-driven research and developing hypotheses, what really motivates my work is mentoring upcoming ornithologists. So throughout my time at Cornell, I've mentored 19 field assistants. So this is a picture of us from last field season. And it's really kind of the spark that I felt whenever I held my first bird in Ecuador. Whenever I hand a student a tree swallow for the first time, you kind of see that spark in their eyes. And all of these students now are pursuing ornithology as a career. So in addition, I'm also involved in a variety of initiatives at Cornell to help increase access for younger generations of scientists to successfully get into graduate school. So I'm currently part of an initiative called Consider Cornell, where we bring over 100 undergraduate students to campus, and we look over all of their application materials. So we do mock interviews, we look at their resumes and personal statements, and then help them get into PhD programs. In addition, I am and was very involved with the American Ornithological Society. So I was the co-chair of their student affairs committee for 2 years. And now I serve on the planning committee for the annual conferences. And during these conferences, we try to have workshops for students that are interested in pursuing ornithology as a career. And then finally, something that I'm very passionate about is my identity as a first generation and low income college student. So I am currently the co-president of the first generation and low income graduate student organization at Cornell. And the whole point of this organization, which we call FIGLY, is to provide mentoring to first generation and low income students. And then finally, as my bio said, I'm also really passionate about public outreach and increasing access to ornithology to elementary school kids in particular. So here in Ithaca, I lead a outreach program at a local elementary school where we go and we teach about avian biology and ecology to groups of 5th graders every year. So what we did was we went and we set up nest boxes around the property of the school. And then as depicted in this picture, we are actually able to then show students live birds by having them observe nest boxes. And during the pandemic, this outreach program kind of came to a halt. So we had to come up with a really creative way to bring the birds to the students, even though they were over Zoom like we are right now. So we developed a method where we can actually go out with phones and we can video the birds and then live stream them to the students' computers. So we published this work in the Academic Practice in Ecology and Evolution journal. And we are now in our 6th year of this outreach program. So with that, I want to thank all of the amazing undergraduate field technicians that helped out. Specifically, these 4 students have done honors theses with me over the years. All of the beautiful tree swallow graphics that you saw throughout this presentation were made by my lab mate, David, and all of the funding sources. And with that, thank you so much for inviting me back, and I'd be happy to take any questions.
Yay. So Jen, I'm going to stop recording and then we can take questions. Somebody had their hand up.
Oh, sure, yeah.